lecture covers cellular metabolism, both catabolic and anabolic reactions. And so here we see an overview of metabolism in which nutrients are taken into the cell and through the process of anabolism are put together into making cell components. And then here nutrients are taken into the cell and catabolized to make energy for growth, energy for movement and transport. So again, movement to power the flagella or to build the flagella, and again, to make cell components. The basis of all this is enzymes, essentially. And so here we see a representation of an apoenzyme. Apo just means that it's a non-functional at this point. So it's the protein portion, the inactive protein portion. And so here the enzyme requires a cofactor or coenzyme. And so cofactors are typically a non-protein portion. And so when these come together, the coenzyme typically will fit into the active site of the enzyme, which then allows it to process substrate. And this becomes a holoenzyme or a whole active enzyme. And so just some examples of some coenzymes. So different vitamins, for example, that behave as coenzymes in metabolism. So all the B vitamins, for example, have all these different roles in metabolism of carbohydrates. And principally what they're doing, so imagine this is a B vitamin, for example, one of the B vitamins that fits into an active site of an enzyme that allows you to catabolize carbon sources, for example. So this gives you a little bit about competitive inhibition. And so in this particular case, in panel A here, you see a substrate being binding to the active site of an enzyme and then obviously being processed. But this is the action of enzyme inhibitors. And so a competitive inhibitor would fit into the active site and block the substrate. That would, again, alter the outcome of the enzymatic reaction. Conversely, a non-competitive inhibitor will bind to this extra little site here called an allosteric site that changes the active site and disallows the substrate from being processed. So we've got competitive inhibition and non-competitive inhibition. So a practical example of that I give is rifampin. Rifampin's an antibiotic that interferes with RNA polymerase in bacteria. And so we often use this to treat tuberculosis, drug-resistant TB, and sometimes the drug-resistant TB is resistant to the rifampin. But anyway, here's a representation of the enzyme RNA polymerase in bacteria. There's 4 subunits that make it up, and then a 5th subunit is added to make the holoenzyme. And then rifampin will actually interfere with an active site on this beta subunit that allows it to dock onto the DNA and facilitate transcription. And so RIF actually in this particular case is acting as a competitive inhibitor. The feedback inhibition mechanism is representative of this process here, non-competitive inhibition. And microbes will use this process pretty regularly to control biosynthetic pathways. So here's a substrate that's being processed by a series of enzymes, enzyme 1, enzyme 2, enzyme 3. In this particular case, it's a 3-enzyme sequence. It doesn't have to be, but so the enzyme is being processed into intermediates, and then the intermediates are processed down to a particular end product. Well, you only need so much of the end product, most likely, and the cell certainly doesn't want to overdo it with end products. So as the concentration of end product builds up, it can feed back and bind to the allosteric site of one of the early enzymes in the process. Typically the first enzyme, that makes the most sense. You want to shut down the first one. You don't want a bunch of these intermediates to build up either, and they could be potentially toxic. So the idea is to build up end product and shut down the earliest enzyme in the process, which is typically an allosteric enzyme, and thereby limiting the overproduction or energy waste of some biosynthetic product. This slide looks at hydrolysis of polysaccharides, but this could be any kind of polymer. These could be repeating units of amino acids in a big long protein. In this particular case, obviously, it's repeating units of sugars in a polysaccharide like starch. And so microbes will produce starch digesting enzymes will break these linkages here, and then results in the formation of the production, the formation of smaller mono and disaccharides that can be easily taken up by the cell and ingested. So here's a representation, two different types of ecological outcomes for these enzymes. And so in this particular case, the microbe is producing an enzyme that goes out into the world and chops up polymers that exist in its vicinity. So imagine like a soil bacterium eating some polymers, some fruit has fallen out of a tree, and it's the sugars from the fruit and the rind or whatever are leaching into the soil, and now the microbe has all this access to these large polymers and has to break them down and then take the smaller pieces into the cell. So in this particular approach, the microbe is using the approach of just kind of pumping out massive amounts of extracellular enzymes and trying to get back what it can from it. In this hypothetical example, the microbe has the enzymes tethered to its surface so that it can bind the substrates directly and then bring them in. So this one seems to be a little bit more clean, and this one's a little bit sloppier, because if I'm a microbe right here where my pointer is, I could theoretically eat this guy's products. So this guy made the enzyme, but I can benefit from it by being over here and gobbling it down. But still a lot of microbes do that. So one of the things you would have done in the lab is to detect amylase activity. Amylase is an enzyme that microbes use to digest starch. And so in this particular case, we do a starch agar plate where we put the microbes onto the plate in a heavy inoculum. And the medium contains primarily starch as the main carbon source. And then you can, after letting the cells grow for a day or two, you can add iodine to this starch plate, and the iodine will turn starch, anywhere there's starch present, will turn the plate black, basically. So this microbe right here doesn't produce an amylase enzyme. There's the black goes right up to the edge of the bacterium. But the bacterium still grew because there's other nutrients in there. However, this guy clearly is digesting the starch, as you can see by the halo around the colonies where the iodine couldn't find starch to complex to and make its color change. So in this particular case, now we're looking at redox reactions, extracting energy from nutrients through redox reactions. So the generic is, you have a reduced substrate like this with electrons available to share, and the reduced substrate gives up that electron, is oxidized essentially, and gives that up to a substrate that's reduced. And so that's what you see here. And so that electron transfer is how we turn that into energy. So in this particular case, now we're using coenzymes like NADH. And so we have some kind of a nutrient, a reduced nutrient that is oxidized, but the electrons have to go somewhere. And so they typically are picked up by oxidized electron carriers. So this oxidized electron carrier will pick up that electron from the reduced substrate, and then that'll form a reduced electron carrier. And then this reduced electron carrier can dump its electrons somewhere else and then produce something else that's reduced. So by this process, we can generate ATP.